Hi, I'm Emily Trunnell, a research associate with PETA. Thanks for stopping by my poster. When I was in graduate school for neuroscience, the forced swim test is something that would come up in discussions about as an example of an animal behavioral test that was often used to draw incorrect or at best specious conclusions about an animal's mood. If you're unfamiliar with the forced swim test, it was popularized by Roger Persalt. It's also called the Persalt swim test in the late 70s. So in this test, a small animal, typically a mouse or a rat, also sometimes a gerbil, hamster, or guinea pig, is placed into a cylinder that's been filled with water. Of course, the animal will swim around furiously, try to climb up the sides of the tank, dive to the bottom to try to find an escape, but eventually they will stop swimming and just start floating. Persalt noticed that when an animal was acutely dosed with a tricyclic antidepressant compound, which were used at the time and are still used today, that they would struggle for longer before stopping to float. So this led him to interpret floating as a sign of depression, or what he called behavioral despair, since acute administration of an antidepressant compound would reduce the amount of time the animal spent floating. Since that time, that interpretation has stuck with a lot of people, and the forced swim test has made an appearance in thousands of publications and in FDA investigational new drug applications for antidepressant compounds. Also, since that time, a lot of people have argued that floating is not a sign of behavioral despair at all, that it's actually a positive trait for survival. Animals that stop swimming and float for longer are saving their energy, they're adapting to their situation. Others have noted really the overall concept that this very simple behavioral readout can in no way stand for the chronic subjective emotional state of human depression. So due to this controversy, we wanted to know how often pharmaceutical companies, these are entities whose bottom line depends on turning out successful therapeutics, had used the forced swim test, and if their use of it had resulted in the successful marketing of antidepressant drugs. So we chose 4 very well-known pharmaceutical companies: Abbott Labs, AbbVie, which is formerly Abbott Labs, Bristol Myers Squibb, Eli Lilly, and Pfizer. We looked on PubMed and Google Scholar to locate any published papers or patent applications where they presented forced swim test data. We then went through those publications and identified each compound that was used in forced swim test. We then used these same databases as well as clinicaltrials.gov to follow these compounds through their development to see, had they gone on to be tested in human depression, what sort of happened. So in our research with these 4 companies, we identified 47 compounds that were given to almost 5,000 animals that were used in forced swim tests by these 4 companies. Only 14 of these compounds, which you'll see in the gray table on my poster, 14 of these compounds that reduced immobility in the forced swim test, which means by that conclusion, they should have some efficacy for human depression. These 14 have also been tested in human depression. So then we asked, what did these human tests show? Was the forced swim test correct in predicting that they were efficacious? And are these compounds currently used as successful human antidepressants? The result was that no, none of the 14 compounds are currently marketed as antidepressants and used by humans. Only 3 of them showed to be efficacious for human depression. 3 of them are still undergoing clinical trials. And the remaining 11 compounds, we found that the forced swim test was not predictive of of the efficacy of these drugs for human depression. So that's a definite 11 out of the 14. I'm sorry. So that's 8 out of the 14 that were absolutely not efficacious. 3 are still pending. So that's at least a 57% chance of the forced swim test giving it a false conclusion. That's higher than chance and flipping a coin. Of the 3 compounds that did reduce immobility in the forced swim test and were efficacious for human depression, they are not currently marketed because of other side effects, that most of which were not predicted by the preclinical animal experiments. So this is a very small sample size, 4 companies, 12 out of 47 compounds, but we're doing more work in this area, and we hope to publish this data soon. But if further data corroborate these findings, that the forced swim test is less predictive than chance at saying whether or not a compound could be efficacious for human depression, then we argue that there would be very little reason for pharmaceutical companies to continue to use the forced swim test, little reason for the FDA to consider it as a viable test in new drug applications, or really little reason for it to be used in basic behavioral research. So in the forced swim test, animals are subjected to the terror of near drowning, and many have drowned in these tests due to inattentive experimenters, or in early versions of the test when, before IACUCs, they didn't really have to rescue the animals from the situation. So it's cruel at its base, but it really misleads drug development and in turn doesn't help suffering patients. So if you'd like to learn more about this, I'd love to talk to you about it. You can reach me at EmilyT@peta.org. Thanks so much.